um, and, and then went on to get our master's at Binghamton University with Steve Bohm, who
We also know.
then moved to IUPUI. And Dr. Melón followed him there, it looks like. And she got her degree there. Her dissertation was looking at binge drinking in C57 mice. And then she went on to do a postdoc at Tufts with Jamie McGuire, and has started at Wesleyan University in 2019. Is that right? And
September
is
2019.
beginning her sure to be auspicious career there as an independent researcher. And we're going to hear some of that work today. And again, I'm super excited, and thank you for coming to talk to us today, Dr. Melón. Take it away.
Yeah, thank you. Thanks so much, Paul. Thanks for the opportunity really to talk with everyone. Like Paul said, I started my lab up at Wesleyan University, which is a primarily undergraduate institution, about maybe 4 or 5 months before we went into lockdown. So in addition to sort of being a bit isolated, it's also been tough. I didn't realize that conferences were part of my self-care routine. So it's been tough not getting a chance to interact with people and talk science. So I'm really looking forward to that chance today. I'll preface by saying I tried to make the talk a little shorter, so it's less on the background information and more so on the work that I've done in Steve's lab and in Jamie's lab, and to sort of lead into what I'm doing at Wesleyan, so that we have time hopefully to talk at the end. It's also, I realize I'm going head to head with an historical inauguration, so I really appreciate everyone's time. It's been an incredibly eventful 2021 already. And we're all living through a pandemic that has ill effects that are harsher for some than others. So today I know is representing a glimmer of hope perhaps to some of us. So I'm especially grateful for folks spending some of that time to talk today. And again, thanks, IBANGS. Invitation. Okay, let's get started. So I'm interested in sex and gender differences in alcohol's effects. And there's very robust findings concerning the expression of what we call telescoping, this shorter duration between the initiation of drug use and the diagnosis of a drug use disorder for many drug classes. But for alcohol, that work has been less consistent and is sometimes contested, in fact, that phenomenon. But what is more repeatedly reported in the literature are these sex and gender differences in the sort of long-term consequences of alcohol exposure. I want to highlight this awesome recent review that boils a lot of this clinical work down. And so in addition to these sex differences in the structural impacts of long-term alcohol use, we know that women with higher estradiol levels consume more alcohol than men. And the increase in rates of binge drinking that we've been seeing recently has been driven specifically by this increase in the high risk drinking for women. And so this pattern of drinking has been known to be potentially a growing problem among women and girls. And we've been talking about this for a while. This is an infographic from the CDC from 2013, so over 7 years ago now, talking about binge drinking as this sort of under-recognized problem among women and girls. And so as folks interested in this long-term biological consequences of binge drinking, it's important or vital really to include sex or biological factors associated with sex in our equation. And so for biological systems that are relevant for the long-term consequences of heavy drinking, there are of course many. I like this helpful sort of conceptual framework that fits the pattern of cycles of intoxication and withdrawal into this greater context of addiction. And it's a useful heuristic tool in part because it also links quite nicely what we know happens physiologically across cycles of intoxication and withdrawal, which is the shift for alcohol, at least in its ability to augment inhibition, which we associate with the state of intoxication, right? Many of alcohol's acute effects mirror the effects we see with classic GABAergic compounds that also augment inhibition. So it links that with tolerance to those effects that develop over time, or an increase perhaps in sensitivity to other effects that may be mediated by excitatory transmission. But together it leaves us in the state of excitatory and inhibitory imbalance. And so we've been focusing our interest on the inhibitory side of this coin and alcohol's well-established effects on GABA-mediated inhibition. Okay. So to introduce GABAergic inhibition to the crew, we know that there are two subtypes of so fast inhibitory signaling that's mediated by this GABA receptor, this chloride permeable channel, and that has been the focus of a lot of my work with Steve and then with Jamie. And it's interesting to think about when you're focused on individual differences in sensitivity and vulnerability to alcohol's effects, because it's such a beautifully diverse system. So it's a heteropentameric channel, so 5 proteins come together to form this chloride permeable channel. And we have a wide variety of proteins encoded by a wide variety of genes that come together or can come together to make a functional GABA receptor. And I pulled this figure from a really lovely recent work that's using, that used analysis of Allen Brain Atlas human brain transcriptome data showing this region and subject-specific expression of these GABA receptor subunit proteins that can come together to form that functional GABA receptor. And so in this GABA receptor system, we have sort of 2 general classes. We have the GABA receptor that mediates your classic phasic inhibition. These are the GABA receptors that tend to be tethered, or that can get tethered by synaptic proteins like gefrin. And we call these synaptically located or synaptic GABA receptors, and they mediate that classic phasic inhibition. And then there's a class of GABA receptors that do not adhere or engage that synaptic protein as well. And so they tend to reside in this extrasynaptic space. We call these extrasynaptic GABA receptors. These extrasynaptic GABA receptors have had a long interest from folks in the alcohol field as potential targets for some of alcohol's effects. There are polymorphism, naturally occurring polymorphisms in the some of the partner subunits, in the partner subunit protein for delta that we know alters sensitivity to the motor and coordinating effects of alcohol. We know that when you have a global knockout of this delta subunit protein, you get significant effects on alcohol related behaviors, and that work has been out there for a long time. I see Greg Komanik's face in the squares that I can see, and a lot of that work has come from his efforts. And so for this question of potential sex differences in alcohol's effects, we think that this extrasynaptic GABA-A receptor is particularly relevant, and I'll get into that why. Other interesting things about this extrasynaptic GABA-A receptor is it's sensitive to lower concentrations of the endogenous ligand, as you might expect it to be functional away from where the action is happening, and potentially sensitive to lower doses of ethanol. Some of that work coming from Ishvan Modi's lab showing this sort of shift in this augmentation of this type of inhibition mediated by those extrasynaptic GABA-A receptors when you introduce alcohol to the system at doses where you don't see changes in the sort of phasic form of inhibition. Those extrasynaptic GABA-A receptors are also very relevant, we think, in stress-associated diseases and in disease states relevant to women's health. And so we know that they have increased sensitivity to neurosteroids, or they confer increased sensitivity to progesterone metabolites that are neuroactive. They can be upregulated by corticosterone, and they play an important role in disease states associated with changes or significant changes in ovarian hormones, like postpartum depression and anxiety. Okay. So to study the role that extrasynaptic GABA receptors might be playing in potential sex differences in alcohol's effects, I've been using mouse models of home cage drinking. And so unlike the clinical work where the idea of telescoping is contested for rodent models of home cage drinking, and especially the intermittent models, we pretty reliably see a sex difference in alcohol consumption. And here I'm showing an intermittent protocol where we give the animals access to a 20% unsweetened solution of alcohol 3 times throughout their dark cycle, and each hour is separated by enough time for them to essentially clear the alcohol that's on board before they are exposed again. And what we see is that females are escalating their ethanol consumption at a faster rate than males, and we don't see this for adolescents. And here we're using C57 Black 6J mice. Okay, and so our question then became, could GABAergic signaling, and in particular tonic GABAergic signaling, be playing a role in this sex difference in some way? And we believe so. And to start, we know that GABAergic signaling in the ventral tegmental area is thought to be particularly important when it comes to the reinforcing effects of drugs in general, and including ethanol, as those dopaminergic projections are under control of the activity of local GABAergic interneurons. And indeed, when we pharmacologically manipulate the GABA receptor system in this region, we can manipulate that drinking behavior. Interestingly, when we target specifically that extrasynaptic subtype of GABA receptor using THIP compound with increased sensitivity for those delta-containing GABA receptors at lower doses, we see that we can, in fact, reduce alcohol drinking behavior by selectively infusing that compound into the VTA. what was interesting and exciting to us is that this effect was impacted by the estrus status of the mice. So for animals that were in this estrus phase, we see no effect of manipulating these GABA receptors in the VTA, whereas for the mice that were in this non-estrus phase, we're calling here, we see that we can downregulate alcohol drinking. So mice have this sort of rhythm of changes in progesterone and estradiol and their metabolites. Unlike the menstrual cycle that is weeks long, theirs is cycling across days or hours. And so we have this phase, this proestrus phase, where we see peak estradiol, and this diestrus phase where estradiol is lowest, and progesterone showing this alternate cycle. And so I'm calling these two phases here where estradiol is low, non-estrus, and these two phases estrus. What was really exciting for us, so when we probe further, we find an oscillation of delta mRNA in this region. So when we take tissue from female mice that are naturally cycling and at either, here we focus on estrus or diestrus specifically, we see that at this diestrus phase, when are responding to our compound, this increase in the relative expression of the delta transcript. Interestingly, in the subunit that's associated with that synaptically located GABA receptor, we don't see sort of like a compensatory change. And so this suggests that GABAergic inhibition has this natural plasticity that may be linked to changes in ovarian hormones or their metabolites. But as well, it suggests that, as others have shown, like my postdoc advisor, Jamie McGuire, D.S. Reddy, and others have nicely shown for the hippocampus, for the periaqueductal bray, that there's this imbalance of subtypes of inhibitory signaling, like this increase in tonic inhibition that's not met with any changes, at least in this case, in phasic inhibition across the estrus cycle. And so once I got, and so all of this work came from Steve Bowen's lab during my time there. And once I got to Jamie McGuire's lab, my postdoc advisor, we could follow up on this and determine functionally if we see perhaps a sex difference in tonic current in this region, and whether that could underlie sort of demonstrated sex differences in alcohol consumption. Okay. Okay, and so for this, we first just simply looked at, so ignoring estrus status. So here we're using females that are not cycling. They're sort of isolated, and we try to avoid cues from the males. And so they often give us this sort of acyclic continuous diestrus smear. Those females are showing this greater expression of delta transcript levels than males when we take tissue from this ventral tegmental area. And then we can go in and measure the levels of tonic current across the males and females by doing whole cell recording, so hold the cell at a particular level, and then introduce a saturating dose of this classic GABA antagonist gabazine that's going to non-selectively block all GABA channels. So we get the shift in the holding current that's reflective of those tonically active extrasynaptic GABA receptors that we've now toggled off. And so we see that the shift that we're using to define tonic current is indeed greater in females than in males. And like the estrus status effect, we see no sex difference in the phasic form of inhibition, nor do we see a difference pulled that data off here, but we also don't see a difference in the gamma-2 protein, so which is indicative of those synaptically tethered, phasically active GABA receptors. And so now we're becoming really convinced that the interaction between these extrasynaptic GABA receptors and some unknown factors associated with biological sex may be contributing to sex differences in alcohol's effects. And so now we wanted to know if manipulation of those delta-containing GABA receptors, those extrasynaptically located receptors, if they would result in sex-specific changes in binge drinking or in the consequences of alcohol exposure. And so to do this, we use a mouse that we designed to have LOX-P sites flanking a particularly important region of that delta gene. And so when we introduce a virus to induce expression of Cre recombinase, what we are left with is a non-functional delta protein. And we can show that that manipulation indeed causes this downregulation of tonic, that tonic subtype of.
No, you can come in, really. Maddie.
I think someone is unmuted. Okay, I'm gonna keep.
That's you, Cheryl. You're unmuted.
Thanks. We had a faculty meeting where someone was cursing in the background and they didn't know. So just to avoid that awkwardness, I'll pause. Okay, so so we see that this manipulation indeed reduces tonic current, but what was surprising is that our effects were not, there weren't really any effects for the males. And we think in part it's potentially because there was such a lower degree of tonic current for the males. Interestingly, though, if we downregulate the levels of functional delta protein in the VTA, we get this really nice relationship between levels of our GFP tagged virus particles for the female mice receiving the Cre EGFP virus, but not for the controls and not for males. And that's specifically because for the females that were receiving that Cre EGFP virus, we are reducing the number of B6 mice or floxed mice on a B6 background that are drinking over 1 gram per kilogram in the 4 hours. And we don't see that for males. Of course, the males already have a greater proportion. I don't know if you can see my pointer here. A greater proportion of mice drinking at that low level. We also made sure to follow up, and we don't see that this manipulation is affecting either their preference for sweet fluids or their aversion to quinine solution. And so now this same effect is not seen when we manipulate expression of that gamma-2 protein that's expressed in synaptic GABA receptors that mediate that phasic inhibition. So we use the same tools, except here we're starting with a gamma-2 floxed mouse, and we introduce the AAV Cre and give 3 weeks in each case to get transduction. And we, in this case, for both the males and females, we're having a pretty significant impact on phasic inhibition in the VTA. Our impact on mRNA levels is more modest, but than the manipulation for delta, but there's a lot more of this gamma-2 higher level than delta. But functionally, we are having a clear effect. When we, however, go back and look at the alcohol consumption for mice after this manipulation, we don't see a change in their drinking. And so to pause and sum it up and take a look out to see if there are any questions, we see that females show a greater escalation of home cage alcohol drinking in various rodent models. Female B6J mice have greater delta mRNA levels and greater tonic current in the VTA. We see that downregulating delta-mediated tonic current in the VTA leads to this female-specific reduction in mice consuming over 1 gram per kilogram of ethanol. And we see no such changes after downregulating the gamma-2 subunit that is regulating synaptic inhibition. And so this then makes us wonder if potential sex differences and vulnerability to the long-term consequences of alcohol involve differences in regulation or plasticity of this subtype of GABA receptor in response to alcohol. So I'm going to take a pause there to see if there's any questions, since I can sort of see some folks. And
there's one question from Daniel Goldowitz. Is it known how the sex differences that you see in the B6 mice, how do they play out in DBA mice? You know, given especially in the context that those different strains of mice have different alcohol consumption. And,
yeah.
They're generally less sensitive to alcohol.
Right. I do not know for this question of tonic current in the VTA. I don't know. That's a good question.
And in addition, Pam Metten also points out that DBA mice are actually more sensitive to some of the classical negative effects of alcohol, like withdrawal CTA, withdrawal severity. So it's an interesting sort of opportunity potentially to dissociate some of those behavioral effects, especially looking at delta.
Yeah. That's a really great point, for sure.
And have any of the transgenics been put on any other inbred backgrounds other than C56 or C57? Excuse me.
For this, no. Of course, the classic work with the global delta knockouts they have, and you do see difference effects if they're on a 129 background versus a B6 background, but we've not explored that at all with ours.
Those are the only questions that we have.
Okay, sorry. Okay, great. I'm gonna. Cool. So now we wanted to evaluate whether we would see potential differences in plasticity of that delta-containing GABA receptors in males and females after alcohol exposure. And so to start, we started with the females and using that intermittent drinking model. The cool thing about it is, in addition to the escalation of intake, you see really nice changes in behavior after just the 2 weeks of drinking. And here we're looking at anxiety-like behavior or exploration, willingness to explore, really, in the light dark box of one mouse that had been drinking water for these 14 days in the same, manipulated in the same way, except they weren't given alcohol in those tubes. And on this side, you see a mouse that will never fully explore this side of the light dark box, and that was one of our binge drinking females. So we see in early withdrawal, so these animals were tested after a 24-hour break. This increase in the emergence delay in the animals that were binge drinking alcohol. And so now we can follow up and say, do we see any significant changes in delta containing GABA receptors or in tonic current in response to that exposure? And so we looked at first just generally and did not see any global changes in protein levels. But when we went back and did immunohistochemistry to look at the co-localization of the delta containing GABA receptor on interneurons, and in particular on parvalbumin positive interneurons, we do see a significant increase in the expression of delta. So at first, it did not seem to fit what we would think would be going on, but when we paused to realize that this increase in the capacity for tonic inhibition on these interneurons would mean you could potentially be sort of pulling some of them offline, or at least reducing their capacity to fire, then you would sort of net effect would be an increase in excitability, which does make sense. And so here, so we've looked a few places, the hippocampus and in the amygdala, and I've sort of focused on the amygdala because we had a really high co-expression of delta and parvalbumin in this region. And we've had some sort of historic reasons for focusing on interneuronal tonic inhibition in this subtype of GABA receptor, in part because there's this idea that there could be an even greater sensitivity to ethanol on this sort of potentially unique version of the delta containing GABA receptor on these interneurons. Okay. And so we targeted these PV interneurons. Here, it's a global expression of the Cre recombinase under control of the parvalbumin promoter. We crossed that line with our Flox Delta mouse to get parvalbumin specific knockout of the delta protein. We inadvertently also landed with a fair amount of global knockouts. We had some germline deletion because parvalbumin is expressed in sperm. And so when it's on the stud line, you can have a high density of Cre leakage. That ends up helping us in the long run, but it just meant this was a bit of a slower experiment because we wanted to go through and confirm who came from which line. And so when we do this, here we're expecting to see, because so far all of our manipulations have been specific to females. So we're thinking that, oh, maybe manipulation of delta containing GABA receptors on these interneurons has some sort of a unique effect for female sensitivity to alcohol or drinking. And so we were surprised to see that in this manipulation, we actually get an increase in the drinking behavior for our males. We see no difference in ethanol intake for the females, but we think there's just a ceiling effect, since what we're seeing is that this deletion of delta on PV interneurons is causing this increase in drinking. And we don't see a change in, say, their sucrose preference. Interestingly, For both males and females, though, we see a shift in their sensitivity to ethanol. So this is starting to look more like what we know we find with the global delta knockout. So we see for the females, when we inject 1.75 gram per kilogram dose of ethanol, and when we've done other doses up to 3 grams per kilogram, so we're getting a much faster induction of that sedative effect. We see this reduction in their sensitivity to that effect, and you see it here as well for the males. And you also see a dampening of sensitivity to the motoring coordination. So when we do the same manipulation and then put them on the balance beam and measure foot slips, we see this reduction in motor sensitivity to motoring coordination for the PV delta knockouts for both males and females. The males show this striking dampening of sensitivity after drinking. So here, these were animals that went through the 14 days of voluntary drinking. They weren't injected. And so we think that's partially why we're seeing this increase in drinking. It's sort of an increase in the for the males. The females, the wild type females in this case aren't showing the motoring coordination at this day 14, but they've been drinking perhaps so much across the 2 weeks that they might be developing some or showing tolerance that the males wild type aren't. Okay. And so what we were initially interested in was that expression of anxiety during that early withdrawal, since that's where we were seeing these changes in the expression of delta on the PV2 neurons. And so when we put these animals that have had 2 weeks of binge drinking through that light dark box assay, what we see is for both females and males, though we're defining it differently, and we can chat about that also. We see this increase in either percent of distance in the dark side of the box, or this increase in the time it takes them to leave the dark side of the box for the wild type bingers. So in both cases, we're seeing some evidence of anxiety-like behavior, though we are defining it differently for the males and for the females, and just looks different for the males versus the females. So I don't think there's potentially a sex difference in anxiety-like behavior in this case. But regardless of how we define it for the males or for the females, in both cases, the PV delta mice do not show that same behavior that the binge drinking males or females are showing. So for the females, the PV delta mice aren't showing this increase in emergence delay once after the binge drinking. And for the males, the PV delta knockouts are not showing this increase in activity in the dark side of the box. Okay. And so lastly, we asked whether manipulation of delta is going to result in sex-specific changes in binge drinking, or its consequences when we focus, I put the wrong image here, when we focus on the BLA, where we saw that high overlap of delta expression and on these PV interneurons. And so what we see is that the selective downregulation of delta in the BLA, where we see this sex-specific increase in delta expression on the PV interneurons, we, again, like our manipulation in the ventral tegmental area, are only seeing a reduction in drinking for females. And so we think that our local manipulations of interneuronal tonic inhibition might be playing a different role in regulating alcohol drinking and sensitivity to alcohol in females versus these changes, these global changes for that the males are also sensitive to. Okay. And so to sum up, again, we see females showing greater escalation of home cage drinking in various rodent models. We see that the B6 mice have this greater expression of delta, greater tonic current, downregulating that delta mediated tonic current leads to female specific reduction in heavy drinking that we don't see when we manipulate synaptic inhibition. The females also show a unique upregulation of delta immunoreactivity following drinking. That was data that I pulled out, but as I showed the females, but not the males. So we were not able to replicate that change in the males. And we're trying to do it again here in our new location. So I don't want to say it outright, but so far it looks like the males do not show this change in delta immunoreactivity on PV interneurons after the drinking, while the females do. And lastly, downregulating delta in the BLA is leading to this female specific reduction of alcohol intake. And so together we think local manipulation of interneuronal delta GABA receptors may be playing a sex specific role in alcohol's effect and on changes in network activity following alcohol. And so with that, I would love to pause and chat more with folks. And thank you again for your attention. This is the new crew. Not much of what I showed here came from them, but we're working hard on getting back in the lab and excited to show more next time.
Thank you very much. I always clap on everyone's behalf. I'll let anyone that has questions, either I'll ask them on their behalf, or you can feel free to unmute yourself. I think that'll work. But I'm going to take the first question, if that's all right.
Yeah.
I'm just curious about in any of your transgenic manipulations, if there's any change in NMDA receptors at all.
I am certain there are compensatory changes that we have not looked at, for sure. Yeah, that's a good question.
A question from Amy Lasek regarding the VTA. Do you think that it's delta containing receptors in dopamine neurons or local interneurons that are important for the ethanol intake?
Yes, that's a great question. We've tried to, and we'll try again. I tried it at Tufts and wasn't successful to evaluate this. Right now, I'm still thinking that it's on the local interneurons because of the behavioral effect that we saw with the reduction in drinking. So after the GABA agonist into the VTA. So I think that it's on the local interneurons, but I don't have great data to support that right now. And do you think the regulation of delta has to do with yes. So right now we are evaluating progesterone metabolites. So we think that allopregnanolone is mediating both the changes in delta expression. Of course, that's also following up on the work of others. But for this particular story with the interaction of delta and sex and alcohol's effects, we also think that alcohol isn't necessarily interacting directly with the receptor, but rather causing changes in progesterone metabolites. And great question.
Great, thank you. Are there other questions for Dr. Melón? If you do have a question, you can go ahead and unmute yourself. That's fine. If there's no other questions, we could wrap it up and say thank you again to Dr. Melón for a great talk, great start to the new year of the IBANGS seminar series, and launching off a new change in the country as well. So I think I think we all appreciate you giving the talk, and we look forward when your lab is up again and running full steam after COVID has dissipated, and we look forward to seeing more of your work come out. I think it's really exciting stuff, looking at the specific regulation of GABA subunits within specific cell types is super exciting from a neural circuits perspective as well. So I just want to thank you for sharing your work with us today.
Thank you. Thanks for the chance to talk to folks and get feedback.
Thank you very much. I'll finish with another round of applause from everyone on everyone's behalf. And thanks again, Dr. Melón, and have a great day, everybody.
Have a great day. Bye.
Okay.